It all begins with the RTK signaling pathway, otherwise known as the receptor tyrosine kinase. In this kind of signaling, the ligand, which in this case is the epidermal growth factor EGF, binds to the EGF receptor. In doing so, it activates the RAF and triggers a phosphorylation cascade, which activates MAPK kinase. Then it activates MAPK kinase, which then activates MAP kinase in the cascade. So MAP kinase basically phosphorylates cytosolic targets and promotes cell growth and division. Then it enters the nucleus and helps with the phosphorylation of other transcription factors, like CMYC, which helps with the transcription of genes and promotes cell growth and division. Now we head over to DNA synthesis. It all starts with helicase, which opens up the strands. Then primase creates the start sequences, or these little primers made of RNA that serve as placeholders and something for DNA polymerase to latch onto. So the DNA polymerase then starts at the primers and builds DNA strands, which matches the new nucleotides to the complementary nucleotides on the old strand. And DNA polymerase also has the ability to edit and take out the primer and replace it with DNA. Last but not least, we have the ligase, which connects any ends of DNA fragments that aren't connected, that were broken by the DNA polymerase. Last but not least, we have mitosis. During prophase, the chromatin condenses into chromosomes, and the centrioles in the animal cell move to opposite ends of the cell. Protein fibers form across the cell, and the nucleolus disappears. Also, the nuclear membrane breaks down. During metaphase, the chromosomes line up in the middle of the cell, and the spindle fibers attached to the kinetochores coordinate movement. During anaphase, the sister chromatids separate at kinetochores, and the proteins holding the sister chromatids together are inactivated. And they're pulled by motor proteins walking along microtubules, and the poles move farther apart. And the last phase is telophase, which is where the cell is getting ready to have its cytoplasm split. And this is where the chromosomes arrive at opposite poles, the daughter nuclei form, the chromosomes disperse, the spindle fibers disperse, and cytokinesis begins.